Hello, welcome to another video. In this video I'm going to be explaining Mendel and genetics. Now, who was Mendel? Mendel was a monk and he worked to look at the inheritance of different features in pea plants. So he looked at both the colour and the height. Okay? Now you don't need to worry about these specific examples, you just need to be able to use what you know about him to apply it to the situation in the exam. So I'm going to go through an example here. So a short pea and a tall pea. Now, tall is dominant over short. So what we note that down as is tall would be a big T to indicate the tall allele or the tall gene, and short would be a small t to indicate the small plant. Okay. Now what Mendel did was he crossed them over. So he bred the tall with the short and he looked at what he found. So I'm going to draw a diagram to show that. So we have our genetic cross diagram. So we've got the genetic cross diagram, and the short pea would have been small t, so that it was short, and the tall pea was tall and tall. So he crossed them over and he bred them together, and this is what we found. So big T, little t, big T, little t, big T, little t, little t. So what we have there is 4 heterozygous pea plants, as in they've all got a combination of a tall and a short allele, but their phenotype would be tall. All of them would be tall because tall is dominant over short. Now, what Mendel did was he looked at it one step further. He crossed over these plants, so he bred these plants with each other. So what we see is this. So again, drawing a genetic diagram. So we've got big T, small t, big T, small t, because all of these plants are heterozygous. So what he did was he bred these together. So we've got big T, big T, big T, small t, big T, small t, small t, small t. So now Mendel was able to observe what happened in this next generation. So what we'd have is we'd have 2 of them being homozygous, one homozygous dominant, one homozygous recessive, and we'd have 2 being heterozygous. But even more interestingly, let's look at their features. These 3 would be tall. So those 3 would be tall, okay? Because they all have the big T, therefore the dominant tall allele. But this one would be small. And the reason this is particularly interesting for Mendel's time is because they didn't understand anything about genetics. But what he noticed was that the first generation, all of them being tall, but then if you breed 2 tall ones together, you'd end up with a small one. So there must be some factor of inheritance being passed and essentially appearing to skip a generation, even though the feature is there. Okay? Now, they didn't know about genes at this time, so they had to make some different suggestions. Now, the reason Mendel's work wasn't discovered until a long time after his death is because he worked as a monk in a monastery, so quite far away and hard to get his work published, and hard to get that work out there to communicate with other people. Another reason is he was a monk, not a scientist, so people didn't necessarily trust his ideas, and also we didn't have an understanding of genetics, so we couldn't explain exactly how it worked. So they're the reasons why we didn't accept Mendel's ideas until long after he was dead. Okay? So thank you very much for watching. Please like, share and subscribe. Any requests, please leave them in the comments. Follow me on Twitter at Rushcliffe Bio. Thank you.